So our next speaker will be Gina Ogilvie, who will talk about the important issue of alternative dose schedules for the prophylactic vaccines.
Good morning, everyone. While my slides are just getting set up, I want to thank the organizers for inviting me. This is really a follow-up to a symposium we had at the last IPV conference with folks who had were either conducting research or had actually implemented alternative dosing programs in their jurisdictions. And this is my disclosure statement. So we all know the background to this, that there's extensive interest in examining alternative and simplified schedules for the HPV vaccine. And this was generated in large part because of the impressive efficacy of both vaccines. And also I think there's interest in looking from a systems point of view if there's ways to improve compliance both by aligning schedules better with existing health programming, such as school health days on an annual basis, and also just by reducing the number of doses. It also gives us an opportunity potentially to reduce some of the resource implications of current HPV vaccine programs. I want to draw to your attention some presentations that will be happening at the conference. One by Scott Lemontang and colleagues about work that PATH will be doing, and that's on Wednesday, and by Amy Creamer and her colleagues with some follow-up on the longevity of fewer than 3 doses of the bivalent vaccine. So here's just a quick summary. As we know right now of jurisdictions that actually have gone ahead with alternate dosing schedules. And again, I want to draw your attention to some of the supplements that we were provided. The HPV Today has a very nice review of some of the excellent work that's going on in Mexico. So in British Columbia, which is my home province, and I'm very happy to be away from it right now because it's raining every day, so it's really wonderful to be here. We embarked on a 2+1 dose series in 2010, and I'm going to describe the evaluation to help us decide whether or not we need that 3rd dose. The province of Quebec in 2008 started with a 2+1 dose schedule, and we are partnering with them to conduct the evaluation for the 3rd dose, but I'm also going to give you a heads up about an interesting proposal they have for further evaluation. I have here Mexico City went with a 2+1 dose schedule for the quadrivalent vaccine as well. Now, actually, if you look in the HPV Today, you'll notice that they're talking now about a regional, not just a regional program, but actually a national program as well. And then Switzerland as well has moved to a 2-dose schedule this year. So let me just talk a little bit about the program in British Columbia. In September 2010, we commenced a 2+1 dose series with a quadrivalent for grade 6 girls, and they're 11 years old, 2 doses at 0 and 6 months, and then an option for a 3rd dose at 60 months. And that was based on data from a study called BC Gov 01, and then we will be doing ongoing evaluation with a study called Quest. Many of you have seen Simon Dobson present this data on the 2-dose study. So this was a 2-dose study using quadrivalent HPV vaccine, 3 arms. The study group, which was 9 to 13-year-old girls, and that we gave them the vaccine at 0 and 6 months. And then the main comparator group was actually study group number 3, which represents the group for whom we have vaccine efficacy. And I think Dr. Dillner's talk was very apropos for us as we look at what are the relevant endpoints for us. But certainly we went ahead with a 0, 2, and 6 schedule in 16 to 26-year-old girls to compare. We also added a 3rd group, which is our study group 2, which are 9 to 13-year-old girls, and gave them 3 doses. Our primary outcome was antibodies for HPV 16 and 18 at 7 months. What we looked at were the differences in the geometric mean titers of antibodies for HPV 16 and 18 and compared it with an ANOVA. And again, further to Dr. Dillner's presentation, we used the Merck CLIA as our primary outcome. And he spoke and I'll show a little slide, but he spoke about the potential limitations of that. We used what had been established for non-inferiority, which was for it to be declared if the lower bounds of the adjusted 95% confidence interval of the Gmt ratios for 16 and 18 were greater than 0.5. So because a picture says 1,000 words, you can see here, this is actually data not to month 7, but out to month 36. And you can see for all 3 arms of the trial that we continue to have good HPV antibody titers across the board. These are the Gmt ratios. The most important group is comparing group 1 and group 3, which is the red group, and you can see that all of them across the board are above that 0.5 threshold. Of note, because everyone sees it, is the group that's in black. That's comparing actually group 1 and group 2. Not surprisingly, you've got 9 to 13-year-old girls. We're comparing 2 and 3 doses in the same age group. So we were actually surprised that we didn't have non-inferiority for 2 of the subtypes. So you can see we achieved non-inferiority for 2 of the subtypes still by month 36. That's for type 16 and type 11. But by month 36, in that 2 versus 3 dose group in 9 to 13-year-olds, not comparing it to the efficacy group, but 9 to 13-year-olds, it is not non-inferior by month 36 for type 18 and type 6. Again, this is using the Merck CLIA. This is again just a picture just describing some of the concepts that Dr. Dildner described earlier, which are the different measures of the vaccine response. And what we also went on to do was look at actually seropositivity using both the CLIA and the total IgG. This is Simon Dobson's slide, and his favorite thing to say is, all you're supposed to see here is that there's a lot of 100% in the IgG. And that's how Simon describes it, and I actually think that's a great way to describe it. So the key is that particularly when you're looking at the IgG, particularly when you're looking at month 36, you see that there's a very high seroconversion rate. Our favorite friend, HPV 18, even at month 36, you can see excellent seroconversion with the IgG at month 36 across all 3 arms. So the summary is that the 2-dose antibody titers in the pediatric population is non-inferior to the 3-dose titers in young adults at month 36. And let's remember, those young adults are the women who we know the vaccine is 98 to 100% efficacious in, and we have 2 doses at month 36 that are non-inferior to that group. We used a broad spectrum of assays, and Mel Krashen will be presenting some data here on some of his pseudovirus assays that show strong ongoing antibody response of 2-dose recipients. The antibody decay curves of the 2-dose girls at month 36 parallel those of the 3-dose population. The plateau mirrors that of 3-dose young girls. And vaccine efficacy of 3-dose in 16 to 26-year-olds is 100%, and we've found non-inferiority for 2 doses, implying that that 2-dose schedule at month 36 should be at least as efficacious. And we know that for programs that are looking at alternate dosing schedules, we recognize that these girls are also going to need to have ongoing cervical cancer screening as girls who have received 3 doses. So regardless, cervical cancer screening needs to continue. Because BC Gov just looked at the antibody titers and not at the clinical efficacy, when we embarked on the program, we committed to doing evaluation. And Dr. Garland's talk very nicely outlined the difference between efficacy and effectiveness. And we wanted to make sure that this vaccine, we had immunogenicity, and we wanted to look at efficacy as well. So we had a very compelling need to continue to examine that. So what we've embarked on is QUEST, and the Canadians in the House will know what I'm saying when I do say indeed that trying to get Canadian provinces together is like herding cats. It really is quite something to try to get us to all agree on something. And so this study is a microcosm of our federation. So it's been quite a work to try to convince everyone how to get together to do this evaluation. But what we have committed to doing with the province of Quebec, you'll note that there's some provinces that aren't part of it, but British Columbia, Quebec, Manitoba, Alberta, and the Atlantic provinces have all agreed to pool our resources to look and evaluate at the alternate dosing programs on a long-term schedule. And the long-term is actually out to 10 years or age, or 10 years post-vaccine or age 19. So the primary objective is to evaluate a 2-dose regimen of QHPV compared to a 3-dose schedule in the prevention of type-specific persistent 16, 18, 6, or 11 in young women at age 19 years of age. We can talk after about why we went for 19 versus month 120, but just back again to the federation type of idea. And I think, again, one of our earlier speakers spoke about the challenges for what endpoint do you use? Do you use the endpoint of CIN23? Do you use the endpoint of persistent infection, cross-sectional infection, immunogenicity? We landed on type-specific persistent infection at 6 months. 6 months. Now, this is a super busy slide, but what I just want you to know is we're going to be gathering a whole bunch of objectives and a whole bunch of data. What we'll be doing is actually recruiting women, and I think it was Dr. Garland who talked about recruiting through Facebook for girls 18. We're planning to recruit about 8,900 girls across Canada. We won't be recruiting through Facebook, but Facebook will be a key part of our retention, as will text messaging, et cetera. And what we'll be doing is recruiting girls who had 2 doses, so it's not a randomized trial, it's a cohort study, and we'll be looking at girls who received 2 doses in Quebec and in British Columbia, and girls who received 3 doses in BC, Alberta, and Atlantic Canada. We'll be enrolling those girls, and we'll be inviting them to provide a self-collected vaginal swab for HPV every 6 months, starting at the age of 15, or 60 months after their first dose. We'll also be inviting a subset of those girls to provide a blood sample to look at antibody titers at month 60 and month 120. So that actually, the reason Simon isn't here for this conference is actually the Canadian Vaccine Evaluation Conference is going on right now, and this is actually going to be launching this week with our recruitment. We received funding from the Michael Smith Foundation and additional funding from province of Quebec. I think what Quebec has proposed, which is also very interesting, is, as I said, they've got an alternate dosing schedule, and they're looking at actually randomizing that 3rd dose in girls and seeing what it offers that way as well. So we'll get some even further data from that, hopefully that study as well. Barbara Romanowski and her crew presented some data at Vigo this year, again providing 36-month data on a 2 versus 3 dose schedule for the bivalent vaccine. And just in the interest of time, a quick review of the approach. They compared 9 to 14-year-old girls to young women aged 15 to 25, with the 9 to 14-year-olds getting 2 doses and the 15 to 25-year-olds getting 3 doses. And they looked at non-inferiority GMT ratios as well. They just used the other end of it and looked for it being less than 2. And they also looked at safety and reactogenicity. And the bottom line, and I'll just show you the data from Barbara, is you can see that they have continued seroconversion and continued non-inferiority out to 36 months in that study as well. So we have similar findings both for the bivalent and quadrivalent for reduced dose schedules out to 36 months. Finally, our colleagues in Mexico have embarked upon a very comprehensive program evaluation, and their preliminary analysis shows that immunogenicity at month 21 of the extended dose, so 2-dose girls and 3-dose girls, shows non-inferiority. This data used the bivalent as well. So their conclusion to date is that the alternative schedule is non-inferiority at month 21. And this is their protocol, and it's very small, but they enrolled approximately 2,000 girls in each arm. So in summary, There's continues to be ongoing international interest in alternate dosing of the HPV vaccine. Data from randomized trials show non-inferiority of both the quadrivalent and bivalent vaccine in girls aged 9 to 12 at month 36 compared to the efficacy population. And I think that's important to note that this is compared to the efficacy population. And I think all of us have an ongoing need and desire to monitor the effectiveness and efficacy of these approaches. And I think we need to heed some of the guidance of Dr. Dillner and Dr. Garland to really figure out what is the optimal outcome measure for these schedules. So thank you very much.